In this video, we'll introduce the concept of protection of the brain that, as you probably can figure out, the brain is a very vital part of our body, and of course it is easily damaged. So we do have a number of things that help to ensure that it will be able to survive at least minor bumps and trauma. So let's have a look. Now, some of the big features that together in working with each other help to protect the brain are the cranial meninges, and that's 3 layers of membranes that surround the brain. The cerebral spinal fluid that bathes the brain fills a cavity. It gives some type of homeostatic stability in terms of the fluid in the cerebral spinal fluid. And the cerebral spinal fluid also helps to sort of float the brain within the cranial cavity, because otherwise the way the brain would be sitting the brain down on the inside of the skull, and so the brain would be pushing up against that skull, and that could lead to potential damage. So it's almost like a bubble wrap also. And then also we have the blood-brain barrier, which we've talked about before, which prevents many substances from entering the brain and protects the cells from real big swings in terms of chemical composition of their environment. So let's start with the cranial meninges. So there are 3 main layers. There's 3 membrane layers, and we're looking at a dense, irregular collagen type tissue. Now, there's also some spaces involved. Now, we have the epidural space, and the epidural space is between the inside lining of the bones that make up the skull and the dura mater. The dura mater is one of these membranes. Then between the dura mater and the arachnoid mater, we have a small space, or a potential space. And then between the arachnoid and the pia, we have the subarachnoid space. So we have the 3 membranes, which are the dura, the arachnoid, and the pia. Above the dura, we have the epidural space. Between the dura and the arachnoid, we have the subdural, and then we have the subarachnoid. Now, let's just talk a bit about some of these. So the epidural space is between the inner surface of the bones of the skull and the outer surface of the dura mater. Now, when we say space, it's not usually a space, and this is this concept of a potential space, that usually the dura is actually pushed up right against the inside layer of that bone. And what a potential space is, and I know it's going to sound weird, a potential space is not a space until it becomes a space, and that kind of sounds a little odd, but what that means is it has the ability to become a space. So if there was blood leaking out and blood got between those 2 features, it could actually form a space. Now, it's as I said, it's only a potential space, as the dura is tightly bound to the bone. And there is, it does allow for the passage of some blood vessels, but it's not a recognizable space in most cases. Now, the dura, or dura mater, is the outermost of the meninges. It's the thickest and most durable. That's what dura means. It means tough, right? So it's a double-layered membrane composed mainly of collagen fibers with a few elastic. Now, the dura actually we can consider as 2 different layers within the dura. Now, most of the time, these 2 layers are actually fused together and create one inelastic membrane. There are a couple places where the 2 layers of the dura are actually separated, and we'll have a look at those. And these are associated with something called the dural sinuses. So what are the names of the 2 layers of the dura? There's a periosteal, which is the outer layer, and it's attached to the inner layer of the bones of the cranium. It sort of functions as a periosteum, and there's extensive blood supply in the epidural space. We also have the meningeal layer, which is the inner layer, and it's avascular, and it's the layer superficial to the arachnoid matter. So really, when we look at the dura, we can consider it having 2 layers. Now, there are a few spots where there's something called a dural sinus, and this is where the two layers are split apart, and they form these venous channels. So these dural sinuses are areas where we have venous blood. They what's their purpose? They help to drain CSF. Now, we haven't talked about CSF yet, but cerebrospinal fluid is really a derivative of blood. It's not the same, but the components of it come from the blood. It circulates through the ventricles and through the various spaces, and then it's returned to the blood in these venous channels. So they drain CSF and they deoxygenate blood from the brain's extensive network of veins. It's also where the meningeal dura folds over itself and courses between brain structures. So we're going to see there's some spaces where the dura actually sort of has itself wedged in between certain structures of the brain, and we'll see that. So these dural folds, we'll give them the name. There's the falx cerebri, and this is a longitudinal fissure, and this separates the left and right cerebral hemispheres, and within that we find something called the superior sagittal sinus, which is really large dural sinus, and that's sort of the superior aspect of the falx cerebri. Now we'll see pictures, so it's going to make more sense, but I want to introduce the terms. We have the tectorum cerebri, which is a partition between the cerebellum and the occipital lobe. And then we have the falx cerebri, which is a partition between the left and right hemispheres of the cerebellum. So what are these? These are really sort of these dural folds that basically, as I say, wedge themselves in between structures of the brain. Now, let's have a look at we're going to go to a picture and have a look. So, before we go too far, so here we are, and we can see that we have the bone of the skull, and here we have, right here, it is the meningeal dura. So you can see here it's fused, and most of around here you see it's fused, but they've reflected it back here, so you can see the periosteal dura and the meningeal dura. And then you can also see below that is the arachnoid below it. And this is sort of one of those regions where I'm talking about where the dura wedges itself in between parts of the brain, and this is the falx cerebri. And you can also start to see some of these sinuses. So these are venous blood filled sinuses that are either collecting deoxygenated blood from the brain, or in addition, they are collecting cerebrospinal fluid and returning that fluid and back into the blood, because CSF, the fluid, came from the blood. It's a derivative of the blood, and now it's returning to the blood. Let's have a look here. Now, this is really tough to see in many two-dimensional diagrams. So certainly if you can look at some three-dimensional anatomy diagrams, you can get a better sense. So these are part of the things like the falx cerebri that goes in between here, and we have the tentorum that are coming in from the side. Let's go back and start talking about some of these features. So we have talked about the folds, and now let's go on to the sinuses. So I have a subdural space, and the subdural space is filled full of this serous fluid, and it's deep to the dura and superficial to the arachnoid. And we can have some veins running through here that drain blood from the brain. And then below that, I have the arachnoid. And that's the second meningeal layer deep to the subdural space. And it's thin, it's web-like, it's dense, irregular, collagenous tissue with a little bit more elasticity within it. Now, associated with this, we have the arachnoid trabeculae, and these are little collagen fiber bundles and fibroblasts, and they help to hold in place or anchor the arachnoid to the deep pia mater. Also associated with the arachnoid, we have these things called arachnoid granulations, and they are also known as the arachnoid villi. And these are little bundles of the arachnoid that actually poke through or project superficially through the dura mater into the dura sinuses. Now, this is going to become important when we start talking about CSF, how it's made, where it moves to, and how the fluid gets back into the blood. It's through here that the fluid of the CSF will re-enter the bloodstream. And then I have the subarachnoid space, which is basically has a lot of major blood vessels, and it's filled with CSF. Now let's look at the deepest of the meninges, and that's the pia. So it's the deepest, and it's the only one in contact with the brain tissue, and it really does follow the contours of the brain, and it covers every sulcus and every fissure. It's almost like it was shrunk wrap on it, and it's in many cases people would see it and not even recognize that it's there, because it really follows that outer surface. And it's permeable to substances in the brain's extracellular fluid and the CSF. And this allows substances to move between fluid compartments and helps to balance concentration of different solutes found in the fluid. So we've looked at this diagram. Now let's go here. This one we can start to see things a little bit better. So here we have, of course, that's the cortex, that's the gray matter. We have the white matter of the brain. This is, we're looking at the superior aspect of the skull here. And of course, this is one of the hemispheres of the brain, and here's the other hemisphere. And we can really start to see that here, the dura does look like it's just one layer, right? If you were to see it, you would assume it's kind of one layer, but we already know that it's made of two layers, the periosteal and the meningeal layer. And in a few spots, the two layers actually split. And that splitting allows for sinuses, like the superior sagittal sinus. Now, as we're looking here, we can see that this is the dura, and then we have a little fine space here, which is the subdural space, and below that, we have the arachnoid, and then you see here are those little fibrous bits, right? And down here is the pia. So you can see in the subarachnoid, that's the arachnoid, that's the pia. In the subarachnoid space, there's actually a space where we do have blood vessels that go through it, and cerebral spinal fluid will actually circulate through here. Now the cerebral spinal fluid, as I said, it comes from the blood and it has to be returned to the blood, the fluid portion. And it's through these things, the arachnoid granulations, that most of the fluid of the CSF re-enters the bloodstream. And we can see these arachnoid granulations right here. Now, as we mentioned before, there are some areas where the dura goes down and separates structures or wedges itself in. And of course, here's one right here. And this is the falx cerebri. And you can see how it comes down in here. And actually, you can see how it splits open here too to allow for a dural sinus down here. Okay, so that's an overview of the meninges and some of the things such as the arachnoid granulations, some of the things like some of the falx that we have here, and sort of their typical location. We're going to end it there. Now make sure you get into the book, you look at the diagrams, you read the book, watch some of the videos. You can go into the 3D anatomy videos, and I think they highlight some of these things also. Okay, we'll see you in the next video.